All right, so some other factors that we need to think about when selecting drugs is the fact that if you are going to take an antimicrobial drug or specifically an antibiotic, that our body is full of natural microbiota and your resident flora that are there to help you keep your body healthy, to help with your digestion, to keep your skin healthy. And so when you select and you take an antibiotic, it's not necessarily that these organisms are safe, right? So the type of drug you take might impact your natural biota as well, right? And not just be specific to the type of pathogen. So again, that's causing the infection. So again, you want to try it and have a narrow spectrum antibiotic that's only going to work on what you're sick from. But in reality, that's not how our drugs are going to work. No matter what, when we're taking a broad spectrum drugs, we're going to kill both the infectious agent and we're also going to kill off some of the beneficial species that you have within your body. Right? So what this can lead to is a super infection or secondary infections, right? So if you think about your natural biota as having sort of this balance, right, where they keep each other in check, they're competing for resources, they're trying to keep the numbers under control. Your immune system is always kind of like monitoring them as well, right? If you are to bring a drug into the mix that's going to kill off, it kind of disrupts the balance within your body. So it can lead to other infections arising from eliminating those healthy bacteria. So the most common example that most people are familiar with is that if you are to take a broad spectrum antibiotic, if you have an infection in your woman, right? Like say you have a urinary tract infection. Well, when you take a broad spectrum antibiotic for that, it can kill off the bacteria in your vagina that are competing with yeast for resources, right? And sort of keep the yeast in check. And, when that happens, then it can lead to a yeast infection, which is, now a secondary infection, one that's resulting because of the drugs that you're taking. So common thing that women have to watch out for when they're on taking antibiotics is that they are at a higher risk for developing a yeast infection. All right, so how do we decide what drug you're going to prescribe? There's a lot that goes into this, right? Ideally, we would like to know the microorganism that's causing the infection. You then have to know what is that microorganism susceptible, what drugs actually work on it. And then, how can we treat the patient? How much, drug can we give them? Can they take the drug? Do they're allergic to the drug? That's how the best we're going to select. So identifying the agent. This seems and sounds easier than it really is. We can, for some types of infections, take like swabs or like bodily fluid specimens. You can take a urine sample, right? You can swab someone's throat for a strep culture, right? And try and determine what is the agent that's causing it. But for the most part, when people have infections, we're looking for secondary things, like maybe they have an elevated white blood cell count, right? We know there's infection there, but we're not necessarily 100% sure what it is. So we just have to make, what is our best guess, and then know that that whatever that organism is susceptible to the drug, right? So if we test and you test positive for a strep culture, you test positive for urinary tract infection, we already know what kind of drugs are going to work on that. So this is the test to actually figure out which bacteria are susceptible to what kinds of drugs, right? So if you have a new type of infection, you can do this kind of test, but this is the test that we're going to do or analyze the results of in the lab. It's called a Kirby Bauer test, antibiotic sensitivity test. And so essentially how this works is that we would take a plate and we would cover that plate with bacteria, and then on that plate we put a disc that has an antibiotic into it. And the antibiotic then leaches out of that disc and spreads across the plate. And the further you get away from the disc, the more diluted that drug becomes, right? Until you get to the point where the drug is diluted so much that it no longer inhibits the growth of the bacteria. So you have this line right here, and we create this circular growth pattern, and it's called the zone of inhibition. Now here you can actually see it actually happening on a plate, and this actually has two antibiotics that are together that we see are having some sort of like joint effects. I'll talk about that in a second. But we can very clearly measure this zone of inhibition. And when we do measure the zone of inhibition, we measure across the entire diameter of the zone, right? And in millimeters. So if we look at the ruler here, we go from 0 to, it looks about 27 millimeters, right? So we have a zone that is greater than 27 millimeters for this, right? Here, I'll show you a table in a second. But this table, this data here shows whether or not we consider that organism to be resistant to the drug. There's an intermediate level, and then or susceptible to the drug. So what this is saying is that if the zone of inhibition were less than 17 millimeters, then we would say that that organism is resistant to that drug. That drug is not good enough to work on that type of bacteria. If the zone is greater than 22 millimeters right here, then that means that the organism is susceptible to that drug. So we're at 27 millimeters, so we're good to go. And in between, that is, we would have that be intermediate. And for every single one, you can kind of see the numbers vary for the different kinds of drugs. So the zone sizes are going to vary on what's effective. So we have a table that will give you like what readings will give you a resistant, intermediate, or susceptible value. Another thing, like this picture, why I have this on here, right, is because a lot of our new drug development, it's difficult to go out and find new antimicrobial compounds. So what they are working on to make drugs more effective are drug cocktails. Like, could you take two antibiotics together, and together they would work better than just one alone? So there's tests that can be done that are similar to this that say, well, if I put these two antibiotics next to each other on the plate, is it going to, inhibit the growth of that bacteria? All right, so the table that you have in your lab manual looks a little bit different than this, but it's conceptually the same. We have the type of drug, and then we have the sizes of the zones and what that necessarily indicates. So if it's greater than 13, 18, 18, 13, right? So these sizes, that full diameter has to be greater than that for that drug to be, or for that organism to be susceptible. If it's less than that, it's said to be resistant, and then in between, they're intermediate. So if we were to take a measurement of Staphylococcus aureus and we got 15 millimeters, well, we look here, 15 millimeters is greater than 13, so we would put an S and say that it is susceptible. Okay, so that's what you're going to be looking at in the lab, is you're going to be measuring those zones and determine whether or not they're considered resistant, intermediate, and susceptible. Now, the other thing that we can do is look at the trends. Like when we look at all of the data, like we've tried this one type of antibiotic on 5 different kinds of bacteria, and every single one of those bacteria is susceptible to that drug. Well, if that drug is effective on lots of different types and groups of bacteria, then that tends to be more of a broad spectrum drug than a narrow spectrum drug, right? Also, we can look at trends in resistance, like seeing how maybe some drug, penicillin specifically, right? There's a lot of things that are resistant to penicillin at this point, just because penicillin has been around and been used the most, right? So we can look at trends like that, like how many different kinds of bacteria are resistant, how many are susceptible. Does that say something about the effectiveness of the drug overall, or does it can help us to determine whether something is broad spectrum or narrow spectrum? So lots of other information that we can extrapolate from that kind of data. The other type of test that's important, and it ties into our Kirby Bauer sensitivity test, is actually looking and seeing how much of the drug is needed to inhibit the growth. So when we look at our disc like this, we just know that there's some line right here where, now the bacteria can grow, right? And I said that all of the antibiotic is here on this disc, and it sort of like diffuses over the surface of the plate to the point where it becomes a weaker concentration out here? Well, there's no numbers that we're measuring, or we don't really know the concentration at this edge, right? But there are different types of tests where we can do that. So this is showing a test, like an antibiotic, it's a Biodisk test, where essentially you can see the different concentration. These numbers represent the concentration of the drug at that point, right? And we grow it on a plate and we see this inhibition of the bacteria all here. And then we can kind of quantify a number of what is the concentration of the drug that's really needed to, inhibit the growth of that bacteria. Another way we can do this is by a dilution series. So a dilution series is when we have just a bunch of tubes with broth in them, media, right? And we put bacteria into all of them. And then into the tubes, we add a high concentration of the drug, right, on one end. Right? And then we dilute. We take a little bit out and we put it in here. And then a little bit out of that one and put it in here. So we're diluting the amount of drug that's in each one of these samples down, and then we can grow them. And we can look and see, all right, at what point do we start to see cloudy growth of bacteria? So I know from this dilution series, somewhere between this 2 to 4 micrograms per milliliter is the dilution of the drug that doesn't work anymore, right? Doesn't inhibit the growth. So we can identify an actual strength or concentration. That minimum strength at which it prevents growth is called the minimum inhibitory concentration. It's the highest dilution of the drug, right? So what is good for this is that we're trying to find how much drug needs to get to the site of the infection, but minimizing the exposure of the person to the drug, right? Which drugs, like we said, no matter what kind of drugs you're taking, could potentially be toxic or dangerous, right? So we want to find that range of what's the size of the drug? How often do you need to take it? How many pills? Every 4 to 6 hours you take 2 pills or whatever, right? This is all starting with determining the minimum inhibitory concentration that's needed. And then a lot goes into the study in pharmaceuticals of how quickly does the drug get removed from the body by your kidney and by your liver if you take it orally, if it's injected, right? So there's a lot that goes into it after it, but it really has to start with what is the minimum amount of drug that's needed to actually inhibit or work on this bacteria, right? The other thing to look at when we're selecting a drug is the therapeutic index, right? So we do want a very wide range between the concentration of the drug that is effective on the infectious agent and the concentration of the drug that's toxic. So way back when we talked about Agent 606, which was kind of this first antimicrobial drug, it was very dangerous because that therapeutic index was very small, right? You gave a little bit of the drug and it killed the bacteria, and just a little bit overdose, and all of a sudden, it kills the person. We want to make sure, if grandma forgets to take her pills and she doubles up and she takes them again, that, it's not gonna kill her, right? So we like to have this wide therapeutic index, giving, room for a little bit of human error so that the drugs aren't super dangerous for people. All right, and that's it. The next part you'll move on to is completing the lab on antibiotics.